I'll invite you to come back to the screen, to come back over here to the screen and let me show you a somewhat more complex application, and I'll talk about a couple other features here. Okay. I'm going to hit next and exit out. Well, let's start this program. Let's save this as another program. Let's save it to PCR. Why would I do that? Well, yeah, I could start from scratch, but why recreate the wheel? I could just click create qPCR, make it a new name. Now on that, I can maintain different copies. It is possible to keep multiple applications open simultaneously. If I save as, I didn't do anything to the original program. It's still there. Mm-hmm. But now I've already selected some labware. If I'm going to use them for the next application, then I could do that. Let's go ahead and do that. Let's go ahead and take let's do a qPCR setup. Everyone, doesn't take too long to write a qPCR program. Let's imagine that got my master mix in that reservoir rack that I had there. I'm going to use all the same settings, liquid detection on. This plate now, I'm going to leave that there. And I'm going to change this volume setting so that all the used positions I know are going to have say 50 microliters in them, because I just pipetted 50 microliters in them. So I don't want the machine to have to scan every single one of those wells to determine that there's 50 microliters. So it'll save time if I just tell it up front that there's 50 microliters there. I'm not actually going to be needing those, so I could delete that and throw on some tips, some 50 microliter filter tips, 50F. I can drop them there, and then I'm going to need a 384 well plate. I could, something. I don't really need that to be cold, but it would be nice if the 384 well plate was cold, right?
Yep.
I could find a 384. Do I have a 384 adapter handy though? Oh, yeah, I do. I guess I do. I'll find an adapter, a thermal adapter for a 384 well plate, and click drag and drop that there. And it wants to call that thermo 2, and then I'm going to go to plates. I'm going to find the PCR 384 well plate. I'm going to find the PCR. And again, it's green. It can deal with this, no problem. Just to demonstrate the gripper, I'm going to go ahead. After I'm done with it, I'm just going to move this plate over there just so you can see the machine move items around, and so you can see the gripper. So I'm going to go into the thermal adapter. It's going to be looking for they're technically a thermal block. There's a thermal block OC, a 96 OC. Click and drop that. I'm going to double check that since this plate will be going from here to there, I need to make sure that that's green as well. It is. So this plate definition can be used in that position as well as this position. And so I'm going to have master mix that's going to be going into the 384 well plate. I'm going to need some samples. The samples are going to be coming from here and going into the 384 well plate. Then we'll have it move and shake, and we'll see that. Okay. Simple enough, right?
Yeah.
Let's go ahead and write that program. It's going to take a little bit. It's going to take a couple more commands and a little bit more thought. Go to the procedure. Sure, we could keep that cold. It would be great if there was no evaporation in my 384 well plate. Let's keep that at 4 degrees. By the way, the thermal adapter can go down to 0 degrees all the way up to 110 degrees C in this position, and the shaker can go from 4 degrees C up to 95 degrees C and shake from 300 RPMs up to 2000 RPMs, but all items cannot be shaken at that high of a speed. So we're going to do that. We're still going to do that. if my first step was moving samples into the plate, then I would need a sample transfer command. And so I would have to drag and drop a sample transfer command into the mix here. And yes, maybe I do want the flexibility of doing up to 96 samples, but I'm going to process a lot fewer today. So I'm going to leave that maximum setting because I want to be able to run smaller numbers. In the sample transfer command, I'm going to be moving just say 2 microliters of sample. I'm going to do it with a single channel 50, a multi-channel 50. I'm going to leave the, I'm going to be pipetting from the samples will be coming out of PCR 96.1. It'll be going into the 384 well plate. There we will be individually pipetting this volume because 2 microliters is not enough liquid to cause a free-falling drop in a reproducible manner if you multi-dispense it. It's far better to pipette small volumes like that. I will select intelligent tool selection, so it'll switch automatically from single to multi when needed, and I will be using filter tips. The pattern, I want to look like this. There's my 384, here's my samples. If I had just one sample, it would be in this position, and that one sample would go there, and the replicate would go there. And then the second sample would go there and there, so on. Again, pattern recognition software makes this incredibly easy. It will carry out your instructions as far as it needs to. So you have coded for the movement into up to 300, up to half the wells. And I'm going to click OK. All right. I do need to at least take a quick look at the defaults and make sure they will apply. Change tip before aspirin from a new source. That sounds like a good choice. Aspirating, default aspiration dispense should be fine. We don't really need to mix it to do a PCR or qPCR. The liquid type will use water for both the single and the multi. So we're good. Now I need to code for the movement of the master mix into the wells. So I have to think to myself, will 96 still apply? Okay, I'm sorry, the reagent transfer, what did I do? I moved the sample, okay, yeah, up to 96 samples being moved. The reagent transfer would be the master mix. I need to change those settings. So say you're doing, you feel like you need 8 microliters of master mix. You want to do it with the multi-channel 50. Select that option. We got filter tips on the deck. We're okay with multi-dispensing that volume. You would have to test that. But minor variations in the volume of master mix really don't have much of an impact on the result. So I'm pretty confident multi-dispensing would work. I'm going to go from the reservoir rack. It notices that there is reset. I was reusing the old program, so I have all the old settings here. Let's go ahead and it will notice. Okay, so this requires a little bit of clarification. A sample transfer command looks at the most recent past number of samples command to determine how many wells will get filled, or how many wells will be moved, how many source positions will be moved. That's what this is looking for, the number of source positions being moved, which is 96. But a reagent transfer command is a little bit different. A reagent transfer command, we already know the number of source positions, 1, or a number of reservoirs that can be paired together and called one same thing. So the only the unknown variable is how many wells will get this reagent. And that's going to change with this number. As this number changes, this number will also change. So that means that, but they're not the same because I'm doing them in duplicate and I'm doing in replicates. So I'm going to actually need another number of samples command here. Now, there are ways of making the machine we can use a calculated thing where we can put in the appropriate we can put in an appropriate calculation here. This is very much like Excel, where you're telling it equals whatever you would tell it what this number is, give it a unique name, and then down here you would say that number, that name times 2 is the number of wells that would be filled. You could do it that way by using the calculated feature, or you can do it manually. If you do it manually, then you need to think ahead. How many wells would you potentially fill? In this case, half of a 384 well plate is what, 186, right? Is that 180? Yeah, 384. Yeah, what's half? 192. 192.
Yep.
Up. $192. All right. And this will also, but see now, this will trigger, if you use the non-calculated method, then this will ask you a question, and this will ask you a question, and you have to be able to distinguish between the two. If you want a different question to be asked, you would simply type that question here in the box. Like when it prompts you for this number, you want it to be basically saying, you know, how many DNA samples in this run? I want it to ask me that question, and when I answer it, it'll adapt it to the number of DNA samples on the deck. When it gets to this number, it will, when it prompts me for this number, it's going to be that number times 2. So I could just type in enter result of number samples times 2 equals. I could put in that. And so that would prompt me to think, okay, how many samples I got? I entered the number of samples, say I'm doing 8. It'll say 8 times 2 is 16. That's the number of wells that are going to get master mix. When I answer that question, it will give me it will adapt to that, to whatever I typed in. I'm going to click OK. So now when I go here, wait a minute, cancel. In doing the reagent transfer, I forgot to change something. My source, I need to redo this. My destination that is not my source anymore. My source is the plate, and the examples. No, the source is the reagent. What am I talking about? The source is the reservoir rack. The destination is the 384 well plate, and the pattern would look like this. From the reservoir rack, if I had just one sample, it would be there. The second sample, the replicate would be there. If I had additional samples, they would be there, and so on. So it would potentially fill up to half the plate. Stops at that number because that's what I chose as my maximum. Click OK. And if I want the machine to automatically adapt to the multi-channel, from multi-channel to single channel when needed, I can click on that. So now once again, you look at the tabs and just make sure that there's not anything weird here. We're not reusing tips. I'm okay with using the same tips for every sample. The default aspiration dispense settings, we're not mixing before or after, or liquid type. Might normally be something different, but we're going to use water settings. I could have used like a PCR premix setting or something like that, but in reality today I'm just working with colored water. And there is a tab down here called rinse. Rinse is a very rarely used thing. If you need to rinse your tips because you're working with radioactive materials, you can have the machine rinse out the tips before ejecting them. I think I'm done. But I did want to move something around, show you some of that and shaking, whatever. So let's go ahead and throw in a transport command. That we're going to be transporting the PCR 96 over to the shaker. And why not just give it a shake? drag down the shaker command, drag down the Thermomix command. Have the mixing on, give it a good shake at say 1200 RPMs for 15 seconds. No sense boring you too long. Set the temperature if you decide to. We won't have it there long enough to get that temperature, so we won't worry about that. I'm going to click check here and see if I've made any gross errors. It didn't find any errors, and click OK. And now I'm going to click play. And once again, do always do a quick simulator with the maximum number of samples, just to see if you might run into a problem if you're using your maximum number of samples. Use all your default settings in a thing. Maybe even add in automatic tool selection if that's an option. Hit next. The number of how many. It will ask you the number of samples. Enter the max, and when you do a simulation like this, 96, 192. How many? 192. Okay. And then I can go ahead and click run because I've got a gripper on there. I've got the two tools here. I'm ready to run on the machine. This is what in simulation mode, it's just going to stay right here though. It's not going to be sending the instructions. Okay, it notices a problem. In quick simulation mode, it says you have 96 tips on the deck, but you'll need 104 in order to do this program the way you wrote it, if you select this max number. So that's something to think about. If I do not change anything, then the machine will still run the program. It'll just prompt me for tips after it uses the first 96. So I'm just going to click OK. In the run, it would give you this method. It would give you this message during the middle of a run that you need to add tips. Click OK. Application ended successfully. Says no problem. I can do this. So we would exit out of the quick simulation, and then we would go back to play, and then we would do a 3D simulation on a limited number of samples. We're going to do a smaller number this time. I'm going to hit next. I'm going to open up and expand that 3D simulation, close it down. Then I'm going to select what do I want to see in 3D simulation mode. I don't want it checking imaginary tips. I may want to see how it's going to do automatic tool selection. I may hit next and answer some questions like how many samples do I actually want to watch? Let's just watch a small number, say 24 samples. No, actually that would be quite a lot. 24 samples in quadruplicate. Well, we're gonna 3D simulate, say, just 16 samples.
Okay.
And that would result in 32 wells being filled with master mix times 2 equals. Okay. Now, when I hit play, when I hit run, I'm going to have to switch over to the other screen to watch it.
Okay.
Hit run, then click over here. I can zoom in and see with it's going to attach tips. It's going to go to my first sample, pipette. It's going to go back and forth, pipette the replicate, throw away the tip. Then I'm going to go to the second one, go to second row. That's right, second row. Pipette and duplicate. By the way, you can speed this up if you want to speed it up. That's real time. We're going to do it a little bit. We want to watch it faster. You can certainly do that. Now it's going to pipette those in multi-dispense mode. So put the tool back, switch for the single channel. Huh. I'm not sure exactly why I moved one. I have to figure that one out. See, move the. Gonna move it to the shaker, shake it, pop up, tell you that everything's successful. Application ended successfully. That's what I wanted to hear.
Okay.
I'm going to hit exit out of here. And I'll leave it up to you. I mean, I guess we might as well go ahead and run it, huh? It won't take but a minute for me to run a few samples in a few minutes, and we'll be able to see the machine move things around. What do you think?
Okay, sounds good.
All right. All right. So once again, over here on the deck, I'm going to substitute that for this. Substitute 50 microliter tips. Eppendorf is always right. Reservoir rack. Plate with thermal adapter here. 384 well plate with adapter. Put that there. I want to hit run and leave the machine, leave that highlighted. And next, I'm going to run a smaller number of samples. I will have the machine check the tips, even though if you have a full box of tips, there's no real neat reason. You can click shut off tip detection. If you're confident that you've got the deck set correctly, you can do that as well. We're going to tell the machine to, I'm going to hit next over here. I'm going to hit next. I'm going to type in how many samples. Let's set this one up to run. We'll do the same thing. We'll do 16 samples. And that will result in 32 wells being filled with the master mix. I'm going to click OK. I wanted to have a reference here so I could look at that, look at the screen and determine, I can make sure I've got the deck set correctly. Okay. Now this time it's going to run very quickly because I've shut off location detection and so on. I'm confident that I have the deck set correctly. 50 filter. I got the PCR 96 here with an adapter. I got the 384 with an adapter. I got that reservoir rack there. The run. Okay. This is let's see it execute the program now. We have it's going to checking the liquid level there for that. Looking for the gripper, looking for the tools. So we're now going to go ahead and pick the 2 microliters from the individual in each pass, going back and forth. Pipette mode. Jettison the tips. Watch out for some more. It's multi-dispensing the 8 microliters of master mix for the 32 wells that it needs. Changing tips. I'm going to switch out. You can see the gripper for the first time. Gripper is attached, and so I move that plate over there. Put it in the shaker. Give that a little 15-second shake.
Okay.
And it will palm out and turn green. Says your program, your application ended successfully. So I'm going to just click OK and exit out of there. Okay.
Okay.
If we could, let's see, if we could move back to the software. I'm just going to very briefly touch on a couple of other commands, so that you at least know some of the other capabilities of the machine. Okay. There is a dilute command here. Like you'll notice that there's a command. There's a command. If you mouse over, it says dilute. This is a pre-programmed serial dilution. This is a serial dilution. If you put a dilute command into the program, it works exactly like the other commands, where you have a source and then you have a destination. All you do is choose where your first sample comes from over here, and on the other side you choose the sequence. So if I were doing that within this sample transfer command, similar to the sample transfer command, if I was coming from a plate and going into a serial dilution within the same plate, I would just simply select the plate as source and destination. And in the pattern, I would say, I want to pick from there, and then I want to go do a serial dilution here, here, here. I'll pick from here and then go here, 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 and so on. So programming is very similar to what you saw in the other commands, but execution is totally different. Because when this is executed, when a dilute command is executed, the machine picks from the first well you selected, goes into the second, spits, mixes, is whatever you preset number of times, aspirates from number 2 to number 3, spits and mixes and does so on, and it does that, and then it changes tips and then goes back for the next sample. So a dilute command just makes programming, rather than having a million commands to program a serial dilution in a plate, you can just do it with one command, and it's really simple to do. On the a number of sample, a pool command is just the exact opposite of a pool command. You're basically taking liquid out of a series of wells and going into a well, and so it works out to be the exact reverse of a sample transfer command. In a sample transfer command, you pick from one sample and you go into the wells, replicates a number of wells. With a pool command, you'll just simply select the source positions and the well that it's going to go to, and keep doing that back and forth. And it's the exact opposite of a sample transfer command. The pool on destination command is the reverse of a reagent transfer command, because remember, in a reagent transfer command you have one source and multiple destinations. Well, you'll select your sources. It will be like your destinations were in the other type in your sample transfer command, and it'll pick from those wells and all go into one well. A pool on destination command is just it's just a very simple way, because you only have to click on a couple wells and say, we're going to go from this plate to here in this sequence. So it's just a simplified pool. You don't have to designate every single source and destination when you use that command. And an empty vessel command is really only used in one scenario, and that is when you want to dry out the wells of a plate. In that one, you have to select both a type of tool, the type of tool that you're wanting to move the sample with. Like say maybe you're using a multi-channel 300, and also whether you want to use the same tool or a different tool to do the drying step. You'll have to designate both. You'll pick where you're coming from. You would probably have to put a reservoir or some waste container on the deck, which I haven't done at this point, but you would designate that, and you will have to select a pattern first. So I don't know, let's just say we're going to be drying out the 96 well plate, and we're going to be going into that reservoir rack. And the pattern looked like, from left to right, we're going to be going here. I'm going to click OK. You also have to designate how you want the emptying to occur. How many times do you want to go back? How much liquid do you want to aspirate? You will sometimes want to over aspirate. When you dry a well, you may want to aspirate more than is actually in the well, and you may do it multiple times. The good news here is that if you do that, the machine will not automatically subtract these volumes, excess volumes, from your sample. It'll record a zero volume, a zero aspiration, a zero volume. You also, this is not something that you would ever change over here on when you're working with a PCR plate, because you do want all your aspirations come from the center. But it is possible with other types of plates to reset positions and have the aspirations occur in different locations around the wells. If you had a large well, a large plate with a lot of different sites that you want to aspirate from, it is possible to set different sites for each one of your aspirations. That's a pretty nifty feature if you're trying to dry out, say, a cell culture plate or something like that. Go ahead and exit out there. We showed you temperature command a little earlier, where you just set the temperature for a block and for the shaker. We did that. There's a mix command where you can mix with tips. All that is you're just telling the machine which plate you're mixing, you're mixing, and it'll attach tips and mix them, and then put the tips in the trash or back in the box. There is a pierce command. If you want the machine to pierce all your wells in a single command, you can do that. There is a wait command used very frequently if you just want to pause for non-deck incubation. There is a set volume command, and that is if for any reason you change the volume of something in the course of a program and you want the machine to understand that you have changed the volumes. Like sometimes I've had people that work with labile reagents, they want to add the reagent in the middle of a run. They will interrupt, they'll put in an intervention command where they'll add liquid to the wells, and then they will need to tell the machine that at this point, the volume is no longer what you thought it was, it's something different. And that's what you use the set volume command for. The transport command, I think we saw with the gripper to move things back and forth. But this is a really interesting command. If you have a really long, complex program, you can actually do you can use an exchange command to do that. You can go here to the work table, and here on the strip, you can copy, say, copy something and paste it here, and save your settings and so on. And if your deck gets completely full, and you no longer, or if you need to substitute something off the deck with something that's on the deck in the middle of a run, you simply place it out here on the park position. And then in the procedure, you tell the machine, at this step in the program, take this item off or take this plate off the deck, and move this plate onto the deck. Move that other plate onto the deck, and put it into that same position where the other one was, something like that. And now the machine will substitute those items. You'll substitute the items, but the machine will be able to do a very complex program. In a production setting where you have many, many, many plates, where you want to run plates all day long, and you don't want to have to reprogram it constantly, and you're not really concerned about it, you can put a program substitution. So at a specific point, when you know this is when I want to do this. I want to replace the tips at this point. I also want to change out the plates and then to let it pick up where you left off. Well, you can do that just by coding it in here with this exchange command, and also putting in set volumes command where you have changed the volumes. When it's executed, it simply stops and tells you to do what you've programmed here. The comment command is really just to put in notes. I could type in, the only purpose for a comment command really is to keep your user, keep you straight while you're writing a program. Like, what does this command do? It's sometimes hard to look at a program after the fact and then determine, like, what was I doing in this step? What was this doing? It was moving master mix, say, but you wouldn't know that just by looking at the command. So if you put in comment commands, other people can come behind you and figure out what the next commands are intended to do, or stuff like that. There is a user intervention command. This is where, like, say for example, you are doing your PCR. You're doing your PCR off the deck. It is possible to put in an intervention command where you tell the machine to remove plate from deck, put on thermal cycler, and then return to deck. You put whatever you type in here, it will stop and tell you to do that before it executes the balance of your program. So that makes it possible for you to incorporate interventions. A user process command, this external process command, the final command, will be your the external process command is exactly like a user intervention command, except for a couple of things. It can, if you select these buttons, one thing in external process command, it will unlock the thermal mixer, so it'll allow you access to things on the thermal mixer that you otherwise wouldn't be able to remove. It will also rescan the location after you're done. So if you substitute something, it will check. It can check behind you to make sure that you've done everything correctly. And that's it. Those are all the commands. It's pretty amazing what you can do with this very limited number of commands, each of which only takes a matter of seconds to minutes to things. So that's your overview of the entire system of the software. Do you have any questions or anything? I can